Good afternoon, dear viewers. I'm your regular tutor, Damilare Akibudi. Today, we shall be considering that aspect of cell and its environment. The conclusive aspects will be considering all other things that happens in the environment of a cell. Okay, now join me in the class. Please do well to subscribe to my channel, Damilare Akibudi, to get more notifications. If you have a question, please drop in the comments and share the video. Subscribe to the channel also. Thank you. So, in the last class, we have taken what diffusion and osmosis is, the experiment of diffusion. In fact, I have also uploaded the experiment to demonstrate diffusion in the solid, liquid, and gaseous states. Okay, do well to check my channel. Today we shall be considering experiments to demonstrate osmosis using non-living material. In the last video where we did cell and its environment, we have done for living materials or organisms to demonstrate the experiment. Now, aim for this is to demonstrate osmosis using a non-living material like yam, potato, and all those. Materials required: 2 tissue funnel, beakers, sugar solution, water, cellophane paper. Now, procedure: you pour equal volume or quantity of water into 2 beakers. Then you cover the bottom of the tissue funnel with the cellophane paper, selective permeable membrane. Then you pour sugar solution into the tissue funnel, A and water into tissue funnel. You see, when we are talking about non-living material, unlike what I have just said, we will be considering tissue funnel, not necessarily using yam or potato, because we will rather say yam and all those are plants, since they can also comply to Mr. Ninja Car. So we'll be considering, when we mention non-living components, we'll be mentioning tissue funnels. That's where we'll be considering. Okay, now the paper serves as the selectively permeable membrane. You then pour sugar solution into the tissue funnel A and B, into tissue funnel B. Tissue funnel B serves as control experiment. Please don't forget the definition of control experiment. It's an experiment done to standardize or to watch what is happening in the real object, in the real subject, in the real experiment. You mark their level, then you immerse the 2 funnels into the beakers containing water. You allow the experiments to remain for about 2 to 3 hours. Now, what's going to be your observation? At the end of the experiment, the volume of sugar solution will rise in the tissue funnel A, while the water level in the beaker will reduce. Water level in the beaker definitely will reduce. Okay, and that indicates that there is a case of either either endosmosis or exosmosis. So that's why it is taken. So to be precise, in that case, since water is getting into the tissue funnel, okay, that is endosmosis. If water is coming out of the tissue funnel, that is what? Exosmosis. At the same time, the volume of water in funnel B and beaker remains at the same level. What is the conclusion? The rise of sugar solution in the tissue funnel A and decrease in water level in the beaker B show that osmosis has taken place. Okay, now what are the differences between osmosis and diffusion? Okay, we have some differences. Okay, diffusion occurs in gases and plants. Osmosis occurs only in liquid Diffusion does not need a semi-permeable membrane, but osmosis needs a semi-permeable membrane. Diffusion occurs in living and non-living organisms, but osmosis occurs only in what? Only in living organisms. Now let's consider osmosis in plants. Osmosis in plants. You know, what do we mean? Plant cells are made up of cytoplasms. They are made up of cytoplasms and nucleus, which are surrounded with cell membrane and the cell wall. They are surrounded with cell membrane and cell wall. The cell wall is tough and fairly elastic. I told you there are some 3 materials that are found on the cell wall. It could be lignin, cellulose, or suberin. If it is lignin, you say it is a lignified cell wall. If it is a suberin, you say it is suberized cell wall. If it's cellulose, you say it's cellulose cell wall. You can't have more than two materials on the cell wall. It's either you have lignin and suberin, or you have lignin and cellulose most times. Okay, now the plant cell is made up of large vacuole, unlike in animals that is small, which is made up of cell sap. Cell sap is simple, is a complex mixture of solutes of high concentration. This causes a net movement of water into the cell from the surrounding, called endosmosis. So the water move into the cell. Since water move into the cell, endosmosis. If water moves out of the cell, we call it what? Exosmosis. It moves from the environment into the cell. Okay, now we consider when endosmosis occurs, it causes the cell to swell. That is when water moves from the medium into the cell. The cell will gradually become larger. Okay, that is what we call, when we get there, we call it turgidity anyway. It begins to become, that's why we say osmosis and diffusion are the two major biophysical processes. Okay, so that it's everything. Diffusion, osmosis is what gives rise to the rest, which we shall consider, be it turgidity, plasmolysis, and all those. We should soon learn this. So, however, it does not rupture because the cell wall, which stretches and does not break. So, osmosis plays the following role in plants. The opening and closing of the stomata is osmosis. All what I'm mentioning now are the examples of osmosis in plants, or importance or principles of osmosis in plants. Number two, movement of water from the soil into the plant via the roots. And if water is getting to the soil from the roots, okay, of the plant, or water is getting to the plant from the soil through the roots, is osmosis. Enlargement of plant cell is osmosis. Okay, turgidity of the plants, like I have just explained, when the cell begins to absorb water, it is what? Osmosis. Now, the following terms are important when discussing the process of osmosis in plants. We discuss turgidity, plasmolysis, and flaccidity. Turgidity, plasmolysis, and flaccidity. What is plasmolysis? Okay, it takes place only in plant cells, don't forget. Turgidity also takes place in plant and animal cells. Haemolysis takes place only in red blood cells, animal cells. Crenation takes place only in red blood cells, animal cells. Okay, so now what's plasmolysis? Is the outward movement of water from the plants, from the living plant cell, when placed in hypertonic solution. When you put it in hypertonic solution, and nutrient, water moves out of the cell. That is what? Then the cell becomes plasmolyzed. Plasmolysis occurs in plant cell. It involves the outward movement of water, because the environment is much more, the solution or the environment, the concentration of the cell is highly concentrated. So water moves out. Okay, now this involves the outward movement of water from the cell, which results in pulling away of the cytoplasm from the cell membrane or cell wall. As a result, the cytoplasm of the cell shrinks. Movement of water out, that is exosmosis. Okay, and the whole cell collapses. The cell is said to have been plasmolyzed. This eventually leads to the death or the wilting of the cell. So we have the death or wilting of the cell when there is what? Plasmolysis. Please don't forget that either endosmosis or exosmosis, both of them leads to the death of the cell. Okay, it is only in an isotonic state that helps us in the survival of the cell. Okay, we'll be considering a very typical case here. The process of plasmolysis is demonstrated in the laboratory using the spirogyra filaments, as you can see on the screen now. Spirogyra filaments. Okay, and how does it go? Here, the cell in dilute solution becomes turgid. Why? Because water from the medium enters the cell until it is fully stretched from the medium. Now here, cell in same concentration of solution, that is isotonic concentration, that is the best. Yes, cell in concentrated solution becomes flaccid. This is in the case of flaccidity. Cell in concentrated solution. If it is in concentrated solution, definitely it will become flaccid. Okay? Yes, plasmolyzed cell. Cytoplasm is pulled away from the cell. Cytoplasm is pulled away from the cell. It is plasmolyzed because the medium where the cell finds itself, okay, is more concentrated. It's more concentrated, and as a matter of fact, when the concentrated stuff like sugar or salt concentration or solution enters into the cell, it makes the cytoplasm to shrink just as you have it now. Then it is plasmolyzed. Okay, now let us move on. Turgidity is a process in which plant cells surrounded by a hypotonic or weaker solution absorbs water from its surrounding to a point where it is fully stretched or becomes turgid, to a solution where it's fully stretched. The content of a plant cell is more concentrated than its environment. The content is more concentrated than its environment, okay, than its surrounding, which contains sugar, salt, and proteins. Protein increase the concentration of plant cell. This causes water molecules to pass through the cell membrane to the cell by osmosis. This inflow causes the cell wall to be able to resist this outward pressure, therefore preventing the cell from bursting. Because the cell wall is much more strengthened in this case. So when it begins to stretch, it does not burst. Okay, it only gets firm. Now we move on to haemolysis. Do not forget I told you haemolysis takes place in red blood cell. Haemolysis is a process of osmosis in animal and red blood cell. Also remember that I told you haemolysis, the two major is diffusion and osmosis, biophysical processes. All others that we have, haemolysis, crenation, turgidity, plasmolysis, flaccidity, they are all the outcome of osmosis, either end osmosis or exosmosis. Okay, like in turgidity, it is end osmosis. Okay, so the water enters into the cell. Now, what do we mean by haemolysis? It's a process of osmosis in animal red blood cell. That is, when the animal red blood cell is placed in hypotonic solution, in the red blood cell, if you put the red blood cell in hypotonic solution, water from the medium enters into the red blood cell, and the red blood cell does what? Bursts. The red blood cell do not have cellulose cell wall nor lignin cell wall. The function of the cellulose and lignin on the plant cell is to strengthen the plant cell. But in animal cell, there is no cellulose, there is no lignin. So anything that happens to the cell, burst it just like that. You will notice that in turgidity, okay, when water enters into the cell, because the cell wall is strengthened with either cellulose or lignin, it does not burst. Sorry, it does not burst. It only gets firmer. Okay, of course, it cannot remain like that. It will soon die off. So, but in haemolysis, the red blood cell does not have cellulose or lignin, so it's not much more strengthened. So it bursts. So the bursting of the red blood cell when placed in hypotonic solution is what we call haemolysis. In a similar condition, when a red blood cell is placed in hypertonic solution, okay, you will have a condition known as crenation. When you have a red blood cell placed in hypertonic solution, water moves out of the red blood cell. So when the environment is concentrated, water will move out of the red blood cell. When the environment, in any cell, please note, when you put a cell in hypertonic solution, water moves out of the cell. When you put a cell in hypotonic solution, water moves into the cell. If in hypotonic solution, you have end osmosis. In hypertonic solution, you have exosmosis. Okay, so the exosmosis process in the red blood cell, when placed in hypertonic solution, is called crenation. It's a condition where a red blood cell is placed in hypertonic solution. Water moves out of the red blood cell causing the cell to shrink. The condition is known as crenation. Diagram shows below hemolysis in red blood cell. Look at it clearly. Okay, this is the red blood cell. This is a crenated red blood cell placed in a hypertonic solution. It shrinks. This is the red blood cell, the red side. This is the swell up red blood cell in hypotonic solution. If it remains in surface membrane, if the red blood cell remains like that, definitely it will burst like this. Hemolysis, red blood cell finally bursts. The outcome of hemolysis. Sorry, the outcome of being in hypotonic solution. Now, we consider differences between osmosis, diffusion, and active transport. Active transport. Process, movement of diffusion, movement of molecules or ions. Condition, high concentrated to low concentration. Additional requirement, down a concentrated gradient. In osmosis, movement of water molecules. Condition, high water potential to low water potential. Additional requirements across a partially permeable membrane. Active transport, movement of particles of substances. Condition for active transport, low concentration to high concentration, and the additional requirement is that it's against a concentration gradient. Energy is what? Energy is required in this case. Okay, with this we've come to the end of the class. We have studied haemolysis, plasmolysis, turgidity, and flaccidity. So thank you. Do well to subscribe, share, and like the video. If you have a question, please drop in the comments. Thank you.